My name is Angelica Ramirez, and today I'll be presenting our first lab report for our microbiology course, talking about hand washing. So, we've all heard the expression, wash your hands for at least 20 seconds. This is very important in maintaining personal hygiene and preventing any spread of infectious diseases. It is a very simple but yet very effective method that reduces the transmission of pathogens from any surface to another and from person to person. The purpose of this experiment is to compare the efficacy of washing our hands with soap and water to rinsing with just water in order to remove the microorganisms from the skin. So the materials needed for this experiment were 4 blood agar plates, 1 agar plate, sterile cotton swabs, sterile saline, hand soap, sponges, and deionized water. We first divided our 4 blood agar plates into left and right sides. The sections of 2 of the blood agar plates were labeled as such, R1, R2, R3, and R4, to indicate the length and method used to wash the right thumb. The sections of the remaining 2 blood agar plates were labeled L1, L2, L3, and L4 to indicate the length and method used to wash the left thumb. Then, we dipped a sterile swab in saline to swab the right thumb of one of our group members and inoculated the section labeled R1. The group member then rubbed their thumb under deionized water for 1 minute. After 1 minute passed, a sterile swab was used to rub the right thumb and inoculated the section labeled R2. The same washing techniques were done for an additional 2 minutes, after which a sterile swab was used to inoculate the section labeled R3. Then the same washing technique was used for an additional 3 minutes before using a sterile swab to inoculate the section labeled R4. Then we used a sterile swab dipped in saline to swab the left thumb of one of our group members to inoculate the section labeled L1. The group member then scrubbed their thumb with a soapy sponge for 1 minute. After the minute passed, the soap was rinsed off with deionized water and a sterile swab was used to inoculate the section labeled L2. The same washing techniques were done for an additional 2 minutes, after which a sterile swab was used to inoculate the section labeled L3. The same washing technique was used for an additional 3 minutes before using a sterile swab to inoculate the section labeled L4. After all of the plates were successfully inoculated, parafilm was used to seal the plates. A sterile swab dipped in saline was additionally used to collect a swab sample from the outer door handle of the women's bathroom on the 3rd floor. Once the sample was collected, the agar plate was inoculated and sealed with parafilm. In this experiment, we followed the lab manual's directions on how to inoculate. For instance, before taking the sample of our thumb, it was necessary to dip a sterile cotton swab in saline before taking the sample. Then we streaked the agar plate in a zigzag-like motion for the best results. And these were our results. Images 1 through 4 on the left side of the slide are displaying the blood agar plates showing the bacterial growth from the samples collected using the different hand washing techniques and washing length. And then image 5 is the agar plate inoculated with the swab sample collected from the outer handle of the women's restroom on the 3rd floor. The top picture shows how the section of the blood agar plate labeled R1 is the portion that was inoculated with the swab sample from the unwashed right thumb of one group member. Here we can see a considerable amount of bacterial growth from various isolated colonies. On the other side of the plate, we can see R2 that was inoculated with a swab sample taken after washing the same thumb with 1 minute with just water. There is a larger amount of bacterial growth and isolated colonies compared to the R1 section. The bottom picture we can see on the left side of the plate, R3, it shows significantly lower amounts of bacterial growth after washing the thumb with water for an additional 2 minutes. The other side of the plate, R4, appears to have no bacterial growth. This section was inoculated with a swab sample taken after washing the thumb for an additional 3 minutes, and there is a drastic decrease in bacterial growth after rinsing the thumb with water for 2 minutes. In the top image, we can see the blood agar plate labeled L1 and L2. The left side of the blood agar plate is the portion that was inoculated with a swab sample from an unwashed left thumb of another group member. We can see a large amount of bacterial growth and isolated colonies in this section, and then the other half of the blood agar plate, section L2, was inoculated with a swab sample collected from the same thumb after washing it for 1 minute with soapy sponge, and we can see no visible bacterial growth on this section of the plate. On the section of the blood agar plate labeled L3 is the portion that was inoculated with a swab sample collected after washing the thumb with soap after an additional 2 minutes. And we can also see no bacterial growth on this section of the plate. However, L4, that was inoculated with a swab sample taken after washing the thumb for an additional 3 minutes with soap. This is after 6 minutes total of washing the thumb with soap. We can see one visible isolated colony in this section. On the clear agar plate that we inoculated with a swab sample taken from the outer handle of the women's restroom on the 3rd floor of HECM, we can see some visible bacterial growth, but it was less than we had expected considering the area and the amount of hand contact that we can see throughout the day. In conclusion, when comparing the results of our 4 blood agar plates, the benefit of using hand soap when washing our hands is clearly obvious. Although there is less bacterial growth between the blood agar plate labeled L3 and L4 versus the plate labeled L1 and L2, the eradication of bacteria takes a longer amount of time in comparison to the samples collected after using hand soap. The majority of the bacteria was killed after 1 minute of washing with hand soap compared to the 6 minutes it took when only rinsing with water. The results collected suggest that washing our hands with hand soap more efficiently reduces the amount of bacteria present in our hands compared to just washing with water. We did notice some errors in our results because in theory, section R1 should have a larger presence of bacterial growth compared to section R2. But section R2 seemed to have a larger amount of bacterial growth. This error could be explained by the accidental inoculation of the wrong section. Another visible experimental error that we can see on section L4 is a single isolated colony. The results of sections L2 and L4 suggest that section L4 should also be free of any bacterial growth. But this was not the case. This error could be a result of contamination of the swab or contamination of the thumb after washing it with the soap for the last time. And that concludes our lab report. Thank you.